In 5.1, we're looking at what is called passive transport. So if you think about what passive might mean, if you think of an active person versus a passive person, it's going to be kind of someone that's laid back, doesn't have a lot of energy, it doesn't take a lot of effort. So that's what we're looking at when we're talking about passive transport. So first of all, a question we're going to try to answer as we go through this section. Sometimes we have plants that are nice and full bloom, the leaves look really great. And then sometimes the plants tend to wilt and get a little unhealthy looking. So we're going to look at why might this happen based on these concepts of passive transport. So first of all, if we look at a cell, a normal healthy cell is going to be nice and plump. We're going to have water collected in a nice amount to keep that cell plumped out, versus when we don't have enough water in the cell, and it's going to kind of shrivel. So here is our healthy plant. It's going to have a nice plumped out cell versus this not so healthy plant that doesn't have enough water. So our cells are going to have to control how much water can go in and out, what's going to keep our plant healthy, and we're going to look in this section at how does that happen. So what happens when we add a concentrated solution to a non-concentrated solution? But first of all, what does concentrated mean? If you think of concentrated orange juice, maybe you buy orange juice in a can and it's frozen, and then you have to add water, or maybe it's some other type of juice, orange juice, grape juice. Concentrated just means all of the sugar particles, all of the ingredients in that orange juice are all packed together real tight. When you add them to water, they break up and spread apart and become less concentrated. So a very concentrated solution is going to be all the particles packed tightly together, and it's not concentrated if those particles are spread out. So if we look at this would not necessarily be very concentrated because all of those particles are spread out very nicely. One thing we have to look at and keep in mind as we talk about all of this is our cell membrane. So in the last chapter, we talked about we've got these phospholipids that make a phospholipid bilayer, bi meaning two. So we've got the first part of the layer up here, and then there's the second phospholipid, so two phospholipids making this double layer. And then we have the inside of the cell and the outside of the cell, and our fluids and our waste and our nutrients are going to have to pass in and out through the cell membrane. Sometimes they can just flow straight through. Sometimes they need a little help with passageways, and we'll talk about those. But it's really important that you're able to picture this cell membrane as we're talking about these processes in this section. So passive transport is going to be transport of nutrients or molecules across our membrane where we do not need energy to make that happen. It can just happen nice and naturally, and the cell doesn't have to do anything to make this happen. So if we look at this picture here, we've got a lot of these little guys, these little molecules on one side, and they're going to the other side to go from an area of high concentration to low concentration. They're trying to even out. Over on this side, they're going through a little passageway and doing the same thing. So there's a lot of the molecules or the particles here, and they're trying to even it out to go to the area where it's not so crowded. So sometimes they can go straight through the membrane, sometimes they need a passageway. Diffusion is that movement going from an area of high concentration to an area of low concentration. And it's going to depend on what particles you're talking about when we're talking about concentration. In this picture, maybe the light blue area is water, and maybe the little purple molecules are sugar. So right now, it looks like in this picture, we've got about the same amount of sugar on each side. So it's all equaled out. But if we had more on one side, then the sugar that's all on one side is going to want to try to move to the other side, some of them, so we are equaled out there. So in this little demonstration, we're going to look at the rate of diffusion and how that is affected by temperature. So basically, diffusion is how one thing spreads out in another. So what we're going to use as our diffusion mechanism will be food coloring. So if I put a drop of food coloring in what we have here is water, we're going to look at how fast does it spread out. So I have 3 different beakers of water. So the first one here is our cold water, and it's measuring 41.5 degrees Fahrenheit. Then I have room temperature water, which is measuring 74.3. And then hot water, which 154, I think, when I pulled it out, Fahrenheit. So I'm just going to put a drop in each one, and we're going to compare how fast those spread out. So I'm going to go for the middle one first. And so we're looking at how fast is it going to basically fill up the water and spread out throughout the water. And we can see that the hot one, those particles are dispersed pretty evenly already throughout the water. Whereas the cooler we get, there's a lot less mixing of the green with the food, with the water. So what this should show us is the warmer something is, the faster the rate of diffusion is going to be. So one other thing we need to look at is called a concentration gradient. And so the concentration gradient is our dividing point of where those 2 different concentrations are, whether it's tightly packed on one side and there's not so many particles on the other, or maybe there's an equal amount. But what is that defining line? So in this picture, the defining line is our plasma membrane, or our cell membrane. So in this first column, you can see there's a large concentration on the top, and there's no particles on the bottom. In the middle one, they have started moving down that concentration gradient. They're trying to move to where they can spread out and trying to even out the amount on each side. And so then finally in that last column, they do have equal amounts of the particles on both sides of that concentration gradient. So equilibrium just means we have the same amount of our molecule on both sides of that division or of that concentration gradient. So diffusion is going to happen across that concentration gradient, but it's also going to depend on the size of the particles. Can they get through evenly? It may depend on what the actual substance is. Is it healthy for that to be on one side or the other? And so if we look in this picture, the spaces that the particles can flow through are large enough that the little blue dots can move, but the green ones cannot. So the green dots can't move and cannot diffuse across the membrane, but the blue ones. Here we're going to look at osmosis, which is a term that you might have heard before, and it's related to diffusion. So with osmosis, we're going to be using the water molecules to get that equilibrium that we were looking at with diffusion, but instead of the particles moving, the water is actually going to move. And I think of it in terms of trying to dilute the concentrated area out and make it the same as the less concentrated area. So if we look at this first picture where it says a hypotonic solution. So in a hypotonic solution, the particles are all located inside the cell, so water is going to rush into the cell to try to dilute those out. And so when all the water goes in the cell, it's going to cause that cell to swell up and create a hypotonic solution. In the middle one, the particles, and this may be salt, it might be sugar, or even in terms of inside and outside the cell. So the water is going to flow in and out, trying to just keep that balance the same. So isotonic is basically the same as the equilibrium that we talked about earlier. So the water is going to flow at an equal balance in and out. In the last column here, the water is going to move outside of the cell because the particles that we're looking at, maybe it's salt water, and there's salt water outside the cell, and so the water that's inside of the cell is going to try to flow out of the cell and dilute those water particles that may be in that salt water solution that are outside the cell. And so when the water moves out of the cell starts to shrivel up, like the little guy we're seeing here. So again, osmosis is the water trying to dilute the high concentration, so the water's moving instead of the particles. So if we look at this, the little red and white dots are salt. So on the first side, there's not a whole lot of salt in that water, but on the second side, we do have a lot of salt. So the water is going to flow from this side to this one to try to dilute that out. And so that would be an example of osmosis. So if we look here, kind of just looking at those hypotonic, isotonic terms again. So hypotonic, we've got the outside of the cell has a lower concentration of solute. And that solute is your substance, maybe it's your salt or your sugar. And so the water is going to try to fuse into the cell. So we can see here, there's lots of the particles inside the cell, not so many outside. So the water is going to try to rush in and dilute those out. And that's going to cause that cell to swell up. Hypertonic, we see there's lots of particles outside. It's very concentrated outside, not so much inside. So the water inside the cell is going to try to go out and then end up causing the cell to shrivel. Isotonic, we've got the same amount of concentration on both sides, so the water can flow equally. Now we're going to talk about a contractile vacuole, and a vacuole is something that we looked at when you built your cell city. So a vacuole is kind of like maybe a package you can think of, but it's an organelle in the cell, and it's going to help move water in and out of the cell. So the water is going to come into the cell, and we've got all this extra cells, so the contractile vacuole is going to swell up, and it's going to gain some of that water, and it's going to move to the edge of the cell, and almost you can think of it like fusing with the cell wall, and then dispersing that water out of the cell. So if we go back to our plants, the plants are going to live in a hypotonic situation and cause the water to move into the cell because there's going to be all kinds of things in the cell that's going to increase that concentration. So the water is going to try to move in. And so then if we look at the blue here is the water in our contractile vacuole, and we have what's called turgor pressure, which is going to be the pressure that the water molecules and the vacuoles are going to exert on that cell wall to push it out. If we don't have enough pressure, then those cells are going to shrink, cause our plant to shrivel up. When they are shrinking, we have what's called plasmolysis. And so you can see the cell is shrinking up because it doesn't have enough water. And so that's called plasmolysis. Sometimes if we get too much water in a cell, and this can happen in our bodies as well, too much water is a problem, and it can cause the cell to burst, and that's called cytolysis. So if we look in terms of the isotonic, hypotonic, hypertonic that we looked at earlier, isotonic, the cell's going to be nice and happy. Hypotonic could cause that cell to swell and bust and have cytolysis. Or we might have that hypertonic where it shrinks up. The next thing we need to talk about is facilitated diffusion. So facilitated means that there's going to be something helping. So sometimes if the molecules are too big to go through the cell wall, or there may be some other reason they can't go through the cell wall, depending on maybe their chemistry, then we're going to have some other way that will help transport them through that cell wall. So one thing that can help is called a carrier protein. And so we're going to have these proteins that are located in the cell wall that are like passageways. And so that they can transport those substances that may not be able to fit through the cell wall by themselves. One type is called an ion channel. And so these are going to allow specific ions to pass through the cell wall. And the channel is going to be specific for the type of ion that needs to flow through. Maybe it's sodium, so only sodium can fit through. The green here is for potassium, so only potassium can go through that. So sometimes the ion channels need to open and close, and those are going to be based on stimuli. And so remember, stimuli is something that causes a reaction. So sometimes the stretching of the cell membrane might cause those ion channels to open and close. Sometimes there might be electrical signals that might affect that, or even different chemicals that might be in the cell can cause those open and close.